We can't hear actually. anything starts, respect and gratitude to our seniors is the most important thing that sustains our life on this planet Earth. As far as human beings are concerned, we have societies and seniors tell us the ways to do it. Juniors find innovative ways to do things. That's my short view of the world. So please have a seat. Don't, please, all of you, please have a seat, please. Seats are empty here. Thank you so much. So without any further delay, I would request, I think we should get on with the main course of the program. So if the thing I have planned is that Professor George Chaldakov, the senior most person perhaps here, who has joined us, would flag off this event. So he is very crucial for my pursuits because he was the first person to recognize and publish my works on an international platform with the specific coinages and the ideas that I was advocating. So, Prabhakar, can you put George Chaldakov on the screen, please? Yeah, please. Sirs, can you please take, then after that we can have you one by one. Professor Senior BHF George Chaldakov. Yeah.
Good morning.
Yes, can you come on video, Senior?
I am seeing auditorium colleagues sitting. You are speaking.
We want to see your face. Where are you? Yeah. Can you get your video on, please?
I'm trying. My grandson helping me.
Try. If otherwise, we can. Can somebody Google and put a face on him? Yeah, can you do that? Please? Yeah. Otherwise, Senior, please.
I thought you have some technical problems.
No, we don't have any technical problem here.
We have.
Oh, okay. This is.
I inform you in due time. You remember, and still cannot solve the problem.
Okay, no
because
problem. We can hear you. Yeah, we can hear you. So this is Professor George Chaldakov. Yeah, he's a physician, and he'll now flag off our symposium. Senior, can you take on from here?
You listen me?
Yes, we can hear you very loud. Yeah, can you?
You are listening me? So I'm. If you're listening me, I like to say, good morning to everybody, chairman and colleagues. I'm absolutely pleased to flag off.
Senior, can you put
slides? Symposium. You listen me?
Yeah, can you put your slides on, please? Can you share your screen?
Slides.
Otherwise, I can put the slides here. Okay.
Okay, put slides.
We'll put the slides here. Okay. I got the slides yesterday. So this one. Yeah, the one. Yeah. Go ahead, Senior.
Okay. Did you see first slide? How do cells work?
It's on the first slide. It's on the first slide.
Engine.
Yes. Go ahead.
Second slide.
Second slide. Yes. Yes, second slide.
You can see my flagging of a paradigm shift. As a cell biology and medical doctors, I affirm that cell physiology, cell biology has some long-standing unresolved problem. Murburn murburn concept introduced 10 years ago or more by Kelan Murali Marjoy Manoj is in my understanding, is a paradigm shift in cell biology of oxygen. I am happy to address this gathering at this every moment. Next slide. I believe I belong to my affiliation departments, two departments, anatomy. You listen me, Murali? Cell biology and translational stem cell biology at Medical University Varna, Bulgaria. I am editor-in-chief of two journals edited by our society, Bulgarian Society for Cell Biology, one journal since 1992 by Medical Reviews, one contributed a lot in the last 8 years, since 2017, and another journal is since 2009, Adipo Biology. I'm also one of the editors of Cell Biology International. My research interests, translational adiprovascular neurobiology, mostly involved in cardiometabolic diseases, their pathogenesis, and eventual therapy. Next slide. I like to say some evolution of in our universe. You know, Big Bang, 14 billion years ago, the universe dance of quark, protons, neutrons, bosons, etc., elementary particles. Next was brain Big Bang. And next, in our understanding, we introduce this adipose biology as branch of cell biology in last 20 years, fat adipose Big Bang. And now, morally, introduce oxygen Big Bang. Next slide. So, you know, American scientist Thomas Kuhn, he wrote in 1962, the book, The Structure of Scientific Revolution. And one of the main messages of this book is that the new paradigm should always be better, not just different. So Morales Murburn concept is following this message. It is better, not only different. Next, paradigm shift. You know, the symbol of this this scientific revolution. Next. So one of the major things of stopping any Eureka appearance emerging, I call this neophobia. So the title of this slide, you can see, is indeed neophobia versus Eureka. So neophobia is the biggest enemy of Eureka. The reason human science, culture, and ethics do not advance as quickly as technology. Doubtless, we have to remember the French philosopher René Descartes' idea of everything must be doubted. So, you know, American poet Robert Prof used to say, we dance around in the ring and suppose, but the secret sit in the middle and knows. So we are, Murari is opening the dance, and hopefully he reach the, he will discover the secret of cell biology of oxygen. Next. So in brief, you know, more about murburn, murzyme, etc. Oxygen facilitate diffuse reagent formation and this need for powering homeostasis and different sensitive response function in cell. So, in brief, Murburn's concept is a paradigm shift in cell biology and fundamental principle of biological life. When 10 years ago, I and my friend of my Professor Yanni, pharmacologist, who were invited to be reviewers of one of the paper of Murari, we first met him a new idea about oxygen life. So his paper was published in one of Frontiers journal, and we are so much great, delighted of young men being so innovative. And we go year by year, we invite in our journals, they are international journals, biomedical reviews. You saw since 2017, 1, 2, 3, 5, 6 volume of biomedical reviews. Wally appeared in front cover with his illustration, and in Adipo Biology Journal 2019 with another paper, and in my principal cell biology textbook issued some 2 years ago or 3 years ago, I also advocated to the readers, student readers, about this new concept, more about concept of relief. So next slide, boy. So again, I mentioned Robert Frost, American poet, you know, and now I would like to mention French writer and pilot Antoine de Saint-Exupéry. So again, we, doesn't matter. It's Thomas Kuhn paradigm shift. Doesn't matter. This is every moment. Urban is every moment in cell biology. We have to keep in mind that we dance around in a ring and suppose, but the secret sits in the middle and knows from the point of Robert Frost, secret sits. And we have to remember Little Prince. In chapter 1, Little Prince draw this figure. So Little Prince asking people meeting on the street, mostly adults, what is this? All adults. And answer, this is cat. But secret is, you can see next slide. This picture is a boa constrictor digested an elephant. This is not kept. This is not kept. So you see that secret sit again illustrated by this picture of by Little Prince of Exupéry. Next. And so brief history of enlightenment in cell biology, starting from Robert Hooke. 1666, a micrographia shows the beginning of some 4th century ago, the beginning of cell study. Antoine Leeuwenhoek first study with introduced microscope, white microscope. Next, a big enlightenment in cell biology, Gregory Mendel, modern genetics. Next, I mentioned Rudolf Virchow, so the father of microscopic pathology. Think microscopically, he said. Next, Robert Frost, German, Robert Koch, German Paul Ehrlich, and Russian Ilya Metchukov about immunity, and Rosalind Franklin, black lady, uh, state lady of DNA discovery. I put first name because she died at 62 Watson Creek, and Wilkins received Nobel Prize for DNA structure. And Rosalind Franklin was somehow neglected by these three big scientists. My field, my next, I like to mention my teachers, Albert in cell biology, Albert Klot, Romanian American George Pavadi, and next Belgian American Christian de Duc, who received Nobel Prize 1974. And my co-scientist in Rome, Italy, Rita Levi-Montalcini, ROM, she got the Nobel Prize for this conceptually for cell growth factor concept, and particularly she discovered nerve growth factor as first member of neurotrophins protein, a Nobel Prize 86. And next, I'd like to mention Günter Bobel, German American Günter Bobel, for his signal hypothesis, protein topogenesis. He received Nobel Prize 1999. I believe that Kevat Morari Manoj, for his murburn concept of oxygen biology, might be a Nobel Prize candidate within this next, say, 9, 5, 9, 10 years, depending on his continuation of paradigm shift in that field, and depending on how big is Indian lobby to Nobel Committee in Stockholm. Next. So, Kevat Murari Manoj, KWM. We named him Homo ebristicus. So here, 1986 in Stockholm, Professor Rita Levi-Montalcini obtained her Nobel Prize from Swedish King Carl XVI Gustav. So here at her birthday, 100 years centennial celebration in 2009 in Rome, Italy. I was invited by co-scientist Roy, her long-lasting co-worker, and presenting lecture. Here is Levi Montalcini, 100 years old lady. At that time, again, 2009, she was born 1909. So me, close to her. I'm proud I work in her institute of Cell Biology and Neurobiology, and Italian Council of Scientific Research since 25 years now. My principal message to this symposium, especially to KWM, my most cordial wishes to Kevat Murari Manoj, to like Rita Levi-Montalcini, meet Swedish King Carl XVI Gustav, and awarded Nobel Prize for in physiology or medicine. Thank you very much for your attention. Go ahead, Murari, and dear colleagues. I am very pleased again to address this Africa Symposium held in a great country as India. Thank you.
Thank you, thank you, thank you, thank you, Senior. Very kind words. Thank you so much. It's inspiring and encouraging. Thank you so much. Thank you so much. Thank you so much.
My pleasure.
Thank you, thank you, thank you, Senior. I'll get in touch with you. Next, we had actually Professor Venkatesh in line, but he is currently, we have Professor Hirohisa Tamagawa from Japan has joined. Dr. Aileen and Professor Aileen Thatcher has joined. Professor Thatcher is actually from our, she's associated with Amrita. So yeah, so we are expecting Professor Laurent Jaeken to join. He'll be the keynote speaker because Professor Venkatesh is not in. So before we have the talk on murburn concept, I would request our esteemed invited chief guest, Professor Raveendran, to say a few words and address this gathering. Please, can you please, whichever, whatever. Thank you so much. Yes, yes, please do.
Most respected colleagues who are gathered here, and also my dear young colleagues and students who assembled here for this program. I was actually not sure about what to say when I come for a program like this, because Murali is so much associated with me for a long time now. He was doing PhD at the same time when I was doing my PhD in IIT Madras. It was IIT Madras then. And he was in chemical engineering and I was in molecular spectroscopy at the Regional Sophisticated Instrumentation Center. So I have known him like a very, very independent, fiery, sometimes outspoken, and kind of a friend who used to live in the same hostel also, where we used to say we used to have all sorts of fun, which we had in those times. So after a long time that I had a chance to actually meet Murali when we had some get together. And that's when I actually came across the work that he has been doing. And I think I don't have to say anything because an expert like Professor Chaldakov has already awarded you the Nobel Prize. Although it has to be, coming in the later years. And, nature is probably the most intelligent system which we have. We talk about artificial intelligence these days, particularly Soman sir is here. But the most intelligent system is nature. Like when you touch upon any aspect of life. I was talking the other day in another meeting. There are several impossibilities which we see in the normal science. You can see actually nature doing. Like we have organic chemists. I do not know whether there are how many chemistry students are here. But as organic chemists, we always say the entire organic chemistry, we need organic solvents, volatile organic compounds we use as solvents for all the organic chemistry synthesis. Because we have the synthesis, and then you have separation processes, so you have all sorts of things there. So we need volatile organic compounds. But I always used to say that, nature is the best teacher. You look at biology. All complicated biomolecular synthesis is actually happening in water. And most of these things are facilitated by these catalysts called enzymes. So they are very specific. They don't make one chemical which doesn't need. I mean, unless you create stress or the environmental stress and other things, nature doesn't create any compound which it doesn't need. And it will make only that compound and it will make 100% specific, 100% yield. There's nothing like 99% yield or 99% specificity. So, which essentially tells us that nature has intelligence in abundance. And many times what we scientists see is only part of the picture. And that is where even scientists have to open up to new ideas in a better way, because like any other religion, the conventional religious systems, scientists too often behave like religions, in the sense there is a set code which you can't walk this way or that way. But we always have seen that it is those people who walked on both these other sides are the ones who told us the best stories. So actually, when I actually had some brief reading about it, murburn concept, and I must say that I have understood it. I mean, I have long way to go in actually understanding these things, in the sense, rather than just being a curious student looking at the cell biology or some natural processes. But then there are several issues which I have come across where you have explained so beautifully, particularly with respect to the cyanide, the effect of cyanide poisoning. Or I had also talked about something, how some concepts, like I do not know whether on a scientific forum how well it will be received sometimes, like the homeopathy, drugs, how at very small dilutions. It's almost same for cyanide poisoning also. Although it is a poison, it is actually acting at very, very, very low concentrations. So how does it actually inhibit the entire oxygen process that is happening in the system? Is that a correct mechanism? Or how the homeopathy drugs work? Is that true? Because many often we see this as a non-science these days. It's very commonly described as a non-science these days. But I always used to say it's like idli. People say that idli is not good, and all. When you say, if it has survived for 100 years, it's good. And similarly, homeopathy as a medicine, branch of medicine, if it has survived for so many years, and if it has cured diseases, for whatever reasons it is, I do not know what the exact mechanism it is, I will say it works. I don't know how it works. So looking at the way that Murali has explained vision and several other aspects, I actually wrote to actually Professor Soman sir that if what he said I do not know, because I am a student who is set to understand all these things. But I think Professor Chaldakov has already beautifully explained, because he is an experienced expert. He is a very well-known person in that area. And don't take my word for that. Better that we take his. I told Professor Soman sir one day when he talked about starting a biomedical school, that here is one person who probably might get, if what he says is true, 10 Nobel Prizes, not one. Well, I don't want to really go on making statements. Let it happen. Science is a self-correcting process where it gets ironed out. All sorts of disparities or different opinions, disregards, all these things eventually has to go through this process of, and it has to get ironed out in the long run, whether it gets accepted or rejected, or what we do not know, because we are taking off. And I think Murali is quite confident. So I told actually Professor Soman sir, and I am so happy that you are here, because I'll tell you one very important reason is, somebody like Murali now at this age with all these things, if you walk into any of the institutes or university, you will not get a job, because there is no provision for them to actually take him at an appropriate level, giving him a job, because people talking about the number of years of experience, this, that, all sorts of regulatory issues which they bring out, rather than pure merit or something absolutely novel coming out. A very important issue which we are all facing. We come across so many people who are outstanding, but who don't find an entry into the academic system. That's where probably I think a system like this, which could really absorb a meritorious candidate like him, and could we have here in this program today meeting all of us. It's a great pleasure. Really, I wish you all the best. And I'm so happy that I was able to come here and join you. Thank you, Professor Saman sir also. And I have Sankaran sir, all these people we have long associations. I mean, he was there from IIT, he was doing his PhD when we were all there, I think. So, thank you very much. I wish you all the best.
Thank you so much, Professor Raveendran. Thank you so much. Here, I have a cardinal principle in life. When you pursue excellence, one thing that you count on is what you have and what you know. I did for the last 8 years or so before joining here. I was at Kolapulli, my native place. I was at home, basically. And thanks to his recommendation and thanks to his the way he does things, and thanks to the management here, I could get a job. Absolutely right. So now we are able to reach out to the world better because of that. So hopefully this process will continue with great gratitude to Professor Ravi and to Professor Soman. Sir, we'd like to invite our next guest, Professor Venugopal and Professor Govindankutty sir. Govindankutty sir is from my native place. He is my neighbor, actually. So he was a professor of IIT Pkd, our neighboring institute here. So, sir, can you please say a few words? And then Professor Venugopal sir, please think if you could say a few words.
So I think Professor Raveendran has picked me to the post. Whatever I wanted to say, he has already said. To start with the murburn concept, that is mild unregulated burning. The role of oxygen in biological systems, he calls it mild unregulated burning. I heard this from him when he came to IIT Pkd some 5 years back and gave a talk. Then he told me that he is also from Sharnor, Kolapulli. And then I found out during the COVID times, because I was new to that place. During the COVID times, I was taking a walk, because after the opening up, after the lockdown, we bumped into each other in some gully in Kolapulli. And then we found out that he is staying here just next to my township. Then he was saying that he wants to develop this theory and build up a study center. Then I was wondering, and I also told him that building up a center in some place as an unaffiliated person, how do you enter some institute and do that? Murali, you must be in some place where you can do this, make a beginning, and then challenge the other universities. Offer yourself for a higher position, and then start propagating this. But this propagation was going on outside India. We have seen biomedical reviews and reputed journals in modern biosciences are carrying reviews, 15 pages, 20 pages, etc. There is a tough fight for space in these journal pages. So he is getting 15, 20 pages reviews published. Textbook chapters are coming on these. And then I again told him, you must get into the system somewhere and then nucleate your own group and propagate it from a position of strength, not as an outsider from a small place like Kolapulli or Sharnor. I'm very glad that now he once called me and said, I am now at Amrita. And I was very glad. I told him that is what you should do. And from here you must take off. And we heard from the authorities now that he is well on his way to were on his way to take off with this topic. And as he said, may that come true, that he gets the wider recognition outside in the form of a Nobel Prize itself, so that I could say that I knew him at that time and we were all his friends. So exactly the three things I wanted to say in the context of this murburn were said by Professor Raveendran. One is the physical biology of vision, then homeopathy. All of us at some time or the other have used homeopathy medicine. Without knowing it's a branch of medicine, somebody is giving you a medication, you are suffering, you take it. Sometimes you get dramatic effect sometimes. So that is a medical line. And the third thing is the cyanide poisoning at low concentrations. So such things are being explained by the murburn concept. And also this enzyme action, he says more zymes, that kind of concepts. This should get wider publicity in the Indian science scene. I hope a beginning has been made, and Amrita University has started it as the nucleation place in India, and this will really take off. I wish Murali and Amrita Institute, Amrita Vishwavidyapeetham, are the very best in this particular context. Wish you all the best. Thank you.
Thank you, sir. So, Professor Venugopal, yeah, okay, sir. Sir. Okay, sir. Okay, sir. Please. Okay, sir. Okay.
Om Amriteshwarya Namaha. From my side, as a representative of the Amrita Vishwavidyapeetham, I extend a most cordial welcome to all the eminent people from within Amrita and from outside Amrita who have gathered here to this seminar. I hope you have a very informative time during the next 3 days. I'm happy to participate in this function. As Professor Raveendran told, me and Murali also were studying in IIT Chennai, IIT Madras for PhD at the same time. Not only in IIT Madras, in the same department. I was also in the chemical engineering department. He was also in chemical engineering department. The only difference was he was working in the biotechnology laboratory, whereas I was working in the heat and mass transfer laboratory. And they were actually in separate buildings. I have to also confess my ignorance about the topic of the seminar. Although I have always been a teacher of the subject of thermodynamics and heat engines. And I went through the pamphlet and tried to understand what is mentioned in that, but without much success. From the knowledge that we engineers have about an engine is something that converts energy from a useless form to a very useful form. To be more specific, heat is considered to be most inferior type of energy, and all other forms of energy like work, electricity, they're all higher quality energy. And heat also, as temperature rises, becomes higher and higher in quality. So engineers have different mechanisms. like we have defined some heat engine cycles where a medium goes through some system where some heat transfer is there. Then it goes through an internal combustion engine or a turbine or something, and then rejects some heat and completes the cycle. In the process, heat is converted to work. We also have engines where energy in the form of potential energy in a dam is converted to work, or potential energy of the wind is converted to work. So all these are our concepts of engines. And I have always understood, and not only that, actually burning is defined in engineering as a reaction involving oxygen, and it is an exothermic chemical reaction where oxygen reacts with something to release energy in the form of heat. And that is the basis of all heat engines. So I always understood that all these carbohydrates, etc., which we consume go to the cells, and the oxygen that we breathe in goes through the lungs and gets into blood, and blood comes into these cells, where the oxygen reacts with these materials and produces energy in the form of heat. I don't know, I may be wrong. And the living cells, or that is the most efficient and most complicated engine. We cannot go and see how it works, but there is some mechanism which converts that energy that is liberated to useful form of work. So living beings are able to do some work. That was my understanding. But now I hear there is a paradigm shift, and some new theories and new role for oxygen is defined in the process of work generation within cells. I am only able to confess my ignorance of this matter any further. And I only wish that the prediction of the inaugural speaker, I'm not able to repeat his name, that we'll see a Nobel laureate from our institution in the near future. Wish him and all of you all the best. Thank you.
Thank you, sir. Sir, would you like to say a few words, please? Yeah. And then Professor Soman sir, after that. Huh? Yeah, only, yeah. I will say, I'll introduce you. Yeah, no problem.
Good morning, everybody. I'm extremely pleased to be here with you people. And when I'm a teacher, basically. I was teaching in the university. Now also I teach in Calicut University. So for a teacher, it is important to see the audience. Naturally, I see many of you people are much youngsters. And here, of course, I should say some elderly people are sitting here. And more than 70% of the people are youngsters here. Me, the other side. And I here, as a teacher, I can keep on talking. Now, nevertheless, it's important to say, I will take maybe 3-4 minutes only, but I would like to tell something which I felt, I did not know Murali at all. Yesterday night, I think by 11:45 or so, I talked with him, and Professor Raveendran told me, interesting, it was a voice message telling that there's a very extremely interesting concept, something he told that maybe a paradigm shift in itself in science happening in biology, and we should listen. Venu, it will be very interesting to you to go and listen to such a concept. I got excited because I am deeply interested in science, its methodologies, its procedures, and its outcomes. So I told, sir, I am coming. And now I am here talking to you. And I felt extremely good to be here. And it is really soothing to hear from such an expert, Dr. George, that India can get a Nobel Prize. I am telling India. Why should I say India, not Murali? Of course, it is latent, that in engine's latent heat, so many things we talk. India is going to get a Nobel Prize because we are starving. India is starving for so-called Nobel Prizes. We have very few. Youngsters, my young friends, it is even to hear from an expert that here is a chance to get a Nobel Prize. Here is a concept to get a Nobel Prize. Itself is a big thing, and for that we should give a big applause to Murali. So a third party is telling that somebody is telling who is not connected to, who is not politically connected, let us say, or any other way connected, telling that here is a person who is going to get a Nobel Prize, and here is a concept. It is very interesting as a scientist, and we should look forward. Now, so there are a few things, Murali, I would like to tell you as a senior person. That is, science is not only, science also needs people to understand, people to listen, people to assimilate, and people to visualize what it is. Because scientists or engineers are really in a closed, to me it is the burning in constrained cells or something. That is I have no idea at all here. So your concept has to go to the masses, has to go to the people who know about it and who value about it, about science, its progress, etc. You should explain, because I am sure if you ask very many people about what this concept is, what is its relevance, why it is called a paradigm shift, etc., etc., any nuances, very many people. Now, I heard 1, 2, 3. We are senior people. We follow science. We read things. We understand. We get excited. Now, 3 of us are telling that we do not know anything about it. How can such a concept get a Nobel Prize? Sure, we can. We people are in the universities. We are teaching. We do not have no idea at all. If we do not know these type of things, very many people, of course, there will be very many community will be knowing. So, Murali, this is one of my advice is that please write articles. Please gives in important simple terms what this concept is, how it is relevant, how it is different also from the normal studies. This is one of the things I wanted to tell. Second thing is, I am again, because Nobel Prize is something which, in my view, I feel the moment I hear about Nobel Prize, I get excited because I am a crystallographer. And in 1970s, after 1930s, we had a Nobel Prize in our pocket. Okay, we means the Indians. In 1970, there's a big feeling in the 70s, 75, 75 that one more person from India will get a Nobel Prize. That person is GNR. GNR was the person. Of course, he did not that luck. Of course, as he told, you have already got a Nobel Prize. Now I remember GNR, of course, already has got a Nobel Prize in inverted commas. Then in 2005 or so, again 5 to 10, maybe 2002 to 2007, again India will get a Nobel Prize in chemistry. There was a winning, and there was one more, there was a very famous scientist. Some people were telling this was our undulations in the scientific society that get a Nobel Prize. Now, I'm not going to want to tell explicitly the name, etc., of such person, but there was one belief that. So, C.V. Raman, then GNR, then one more person. Now I hear, really, it is his name, and we feel, all of us feel good about it. So, it has to go to masses. People should understand. People should relate. People should negate also. I am sure no concept is that is all pervading. So it will have its own negations, it will have its own limitations. As science says, Professor Raveendran told is science is ever evolving. So there will be very many things happening. And now I am in my shoes. If I talk now, I keep on talking. Now some other concept in place. I will just stop in one more minute. Many things come to me the moment. For example, simple chemical engines. Is simple chemical engines, is it okay? Can we have a simple chemical engine? The question is that I don't know what is meant by simple here exactly. I understand a chemical as an ordinary person, a chemical engine. Can it be a simple chemical? What does it mean? A simple chemical engine. Can it be a simple complex? This is my idea in my mind. Of course, it is a word only. The simple is a word to express idea. Can it have a simple chemical engine? Now, one more question again. Does a chemical engine negate God? Chemical engine. I came with Professor Raveendran's car. It is let us say it's a big engine. Does it need God for running it? Now, if it is a cell is a chemical engine, and I am a collection of cells, do I need a God for running this? Like that, youngsters think, diversify, think very many things. Let Murali think, diversify. Let him make understand very many things, and let us. There was a word eureka there. Let us look forward for that eureka. Thank you very much.
Now, there were many things said by Professor Raveendran, Professor Sankaran, Professor Govindankutty, Professor Venugopal sir. I would like to address all that, but before that, we have our own Dean, Professor Soman, who would like to put this also into context of the affairs that we do. Basically, we are an educational institution. We run programs. We get students. That's what makes our things move, right? So Professor Soman would just take off from here and would like to say that this murburn is a content of our course, a new course for which I'm getting recruited. I got recruited, I got employed. That reason is a new course called Artificial Intelligence and Data Sciences in Medical Engineering. So Professor Soman would, our Dean, loved, respected Soman sir, would now talk about something that he wants to project at this occasion. We have got an audience, we are going to reach out to people, and so he would like to talk about it. Please, sir.
Good morning and welcome to this symposium. I'm going to do a sort of marketing job. We are running this course, so running this symposium as part of our this new program in A and medical engineering. So I would like to tell a few minutes about this this program. As Prof. Murali, simple reasoning is enough to explain all the normal and anomalous behavior of cells. That is what convinced me. I don't know much about this area. I am the process of learning, and probably we and our students will start applying AI to understand this natural intelligence within the cells. So similar things are happening in AI also. Initially, AI was thought something very big. Okay, but over time now it is simplified like anything. Even an high school student, I say even the mathematics behind that one can be understood by at most +2 level. It's a course from MIT. It's a 2 weeks course. Okay, just 2 weeks. Now 7 lectures is covering the latest of the AI. Covers almost every development last 20-30 years. Okay, simplified to that level. And now I took this material and gave it to our first year students. They are able to understand. So that possibility is there. And there are a lot of examples coming in other universities. This is from I think Berkeley. Okay, 2 course. Course name is Designing Information Device and Systems. But this is a course running over 3-4 months. But this I am electrical engineer. I am telling this course plus second almost covers basic electrical and electronic engineering. Okay, so question comes now: do we need to spend 4 years just to learn? Okay, the required things in current electrical engineering is a question. Okay, so my answer is no. And in this program, we are trying to combine almost all the basic engineering. Okay, AI, computer science, medical engineering, devices, medical biology, and supported by all the core engineering courses. Okay, and it is possible. That is, I firmly believe on this. Okay, other thing. This AI is impacting our two areas. It is impacting maximum. One is education, other one is healthcare. So we need multidisciplinary courses with a strong foundation of computer science. So we are designed this way. This is what it is. So as per the experts there is a lot of disruption happening in medical science. Okay, it can reduce diagnostic cost, little. We can combine AI with modern and traditional this one. AI can reduce treatment. All these are possible now. So we are incorporating all this into our new program. India can become a tourist healthcare tourist spot. And our thing is AI with robotics and LLMs for healthcare, that is support for aging population. Okay, so this robotics is also part of our this new program. Okay, now learning medical sciences. I don't know what is happening here. This. Okay, this now. Okay, no problem. So doctors are taking now 5 years for learning this medical sciences. Okay, 2.5 years for basically about anatomy, physics, and all those things. Now using AR, VR. Okay, we have set up a system there. Okay, you can have a 3D view without any glass. You can have a 3D view of organs and anything about the biology. So learning medical sciences we can do faster with the latest technology. So there also this is possible. Fraction of time, and we have the setup. DNA sequencing. Now, a lot of AI can be applied here for personalized medicine. Chemistry side, there's now a lot of changes happening. The way we learn chemistry. Okay, so organic chemists are embracing AI for faster exploration of the chemical space. Okay, number of chemicals possible as per the chemist is much more than number of atoms in the universe. So how to explore that chemical space will require, probably AI will help for faster drug discovery, and supporting softwares are coming where AI is applied to all the current existing databases. For example, data AI into education for faster active learning and critical thinking. There is something called a software called retrosynthesis. You say what molecule you want. Okay, system will tell now how to produce that one. The set of action your side required, or set of probably reaction. Okay, required for synthesizing that one. Okay, it is in the initial stage, but soon now it will be readily available for now probably any compound you want. And there is a new trend, actually global trend, merging of the medical science, artificial intelligence and biomedical technologies. For example, one in Hong Kong University, 6-year course. Once you finish 4 year, you get an engineering degree. You finish 6 years, you get MBBS degree. And also in one in Italy, same setup. 6 years, you get engineering degree and medical degree. So about curriculum, I already told now we combined everything. Okay, and these students, they specialize in some electives. They can A in medical science, A in CSC, A in robotics, A in communication engineering. All these are possible just within 4 years. This possibility is there. Technology allows that one. It's not magic. Okay, modern technology allows that one. Other thing is the step up credential, micro credential courses. In fact, ICT and UGC want this type of course to be introduced, because already MIT started. Okay, 2-week long courses. Okay, finish now do some projects. Okay, finish all that course. So in MIT, when a student enter he register some department, but he is free to take any. You can go anywhere and take the subjects, and after completing a few number of credits, okay, degree is given according to the weightage where you are focused. Okay, that is the way modern world is moving. Okay, there is no barrier between sciences and engineering, nothing. Okay, so that type of flexibility people are making now. So we have every semester, along with other 4-month-long courses, we are also giving micro credential courses. Each will carry only one credit. Okay, so advantage for this one as something new comes, we immediately put in the syllabus. Okay, so nursing semester is going this way, and supporting technologies. New kind of infrastructure we are. Okay. So to do it, what we are doing is, one is new kind of infrastructure. Basically, we are having now class of thin clients. Okay, the thin client is connected to server. They won't able to use any other access internet and do coding. Okay, in a confined environment. Okay, every class. Okay, so every subject now we are mathematics and coding in every subject. Even biology requires now more mathematics and coding. For subjects that are project oriented, we replace final exam with projects. Okay, there is now every subject final exam that is replaced with the project. So in a semester now every student will be doing 6 projects in groups. Then okay, already told about micro credential courses. So these type of devices are there now. You don't require a lab. Olden days now you have to go to laboratory. Okay, you have to take equipments from there, do the experiments. Now these type of devices you can carry in your pocket. Okay, rather than 1,000 all these devices, now students can learn electronics, communication engineering in no time. Only thing is is basic maths. If it is thoroughly understood, learning all these things won't take time. Okay, they can do under thousands of experiments. Corresponding website is having all the electrical communication experiments. Website is given. just download or now just go through it, use these devices to learn it. Now, of course, now going to lab. Okay, that concept is out. Lab in a pocket. Okay, other thing what is happening now, there is concept, this is analog computer, it is coming back in a different way. Analog computer is conventionally used for solving differential equation, but now what people are doing is they are embedding their learned model in a neural network into this. I don't like this one, I don't require power. 3 watts. Okay, this is enough for it. So learned model, learning don't use this one. You use conventional digital computer. Once learned, you embed into an analog device, 3 watts. So future what is going to happen now, whatever power requirement is now, ambience now it will collect and it will run. So every device now within 3 years become intelligent. Any device or now here everything. So whatever is there. Okay, based on this new technology is happening now. So we have a computer now, this one here, it's from Germany. Okay, this type of robotics and everything can be everything now can be learned quite easily now. Technology allows and very cheap. Okay, anybody can afford it. Okay, other thing what is happening as far as students are concerned, students are learning for examination. Take away that concept. Okay, skill up development should be students' goal. Okay, so for continuous learning, what we are doing is again thin client, connect to server, half an hour or maximum 1 hour class, followed by a discussion, followed by a test. Test will be automatically validated. When you are coming out of the class, you will get the result. Okay, so this we are introducing test and automatic validation in every classroom. So current next semester onwards, we'll be doing it. Okay, so this is very important. Daily basis learn. Don't postpone anything for final examination. Okay, and the classroom itself is lab. That is the new thing, and there is no separate lab. Okay, so our learning pattern or the way we are taught now, everything we should change now. New pedagogy, okay, theory, computing, practical, all together. And there are now new generation books like this. Okay, so actually it's a Berkeley is meant for now +2 students. Okay, by the time you pass that one now, you might have learned all the basics, but now use of computers is a must. But our current in our high school syllabus, okay, for namesake computers are there, but they are never using computer for anything other than watching video. Okay, so this integration is a must. We are learning mathematics, immediately do some basic coding. Okay, maths now, modern maths, especially required for AA. You can't do by hand.
Yeah, yes, okay, thank you.
Do that one. Then new generation, +2. Just +2 itself, we can teach a lot. And this type of books are coming. Because now most of the things in this book now, first part is in Excel, spreadsheet. Almost all engineering now, whatever differential equation comes now, that's all in Excel. Just click and drag, you get the answer. Okay, then MATLAB or no. So that is how. Okay, again, robotics also same thing, because the books are coming that way with theory and practical together. So all can be learned, okay, by anybody, not mechanical engineer alone. Anybody can learn robotics in no time. Okay. So this is about our new course. Thank you.
Yeah, thank you so much, sir. Just for the online participants, are you still here?
Happy. Trying to get this thing here. Is Professor Laurent Jaeken? Hello, Laurent. Can you hear me?
Yes. Yeah, I hear you.
Good. Laurent, please unmute your mic. Unmute your mic.
Mute.
Your mic is muted. I can't hear you.
was not muted.
Okay. Now you're on. So thank you. This is Laurent Jaeken, folks. Please put up a hand for him. He has been. Yes, Laurent, this clap is for you. Yes, Laurent, we'll have you after some time. So people, this is Professor Laurent Jaeken. He has been a Nobel laureate student. Now, since the buzzword is Nobel, that gets people's attention. That's why he's a very seasoned scientist. He has been working on multiple areas. He has written books. So he will talk about himself. We are in the same group now. I will start off with the talk and I'll give it on to him, the main crux for which all of us are here. And you put the the first one, the talk, please. See you in a bit, Laurent. Can you hear me like this? You want me to use the mic like this? Yeah, I'd rather. I don't like it. I like it. Yes. That's great.
Dr. Murali, it seems like Laurent Jaeken's microphone is still muted.
He is not needed now. Oh, okay. Afsal, hi.
Hi, no, I'm just
yeah.
saying that Dr. Laurent's microphone is still muted.
Yeah, he needs to be on the mute. Afsal has joined us from Oman, Qatar. He was my student from. Sorry. He was my student from the days I used to silence all the other. Because I don't want.
Hello, you hear me?
Yeah, you are on, Laurent. After about 10-15 minutes, you will be on. Okay.
Yeah, fine.
Yeah. After And she also said that theories must prove to be facts or be discarded as worthless. Efforts must soon be crowned with success or discontinued. Once we know that something is wrong, we should not stick on with it. That's a fundamental thing that I wanted to say. Next, a discovery must be, by definition, at variance with existing knowledge. In science, the Apollonian tends to develop established lines to perfection, while the Dionysian rather relies on intuition and is more likely to open new, unexpected alleys for research. There are different kinds of scientists. There are mixture of them also. So many a times their agenda might not converge. Sometimes when you have been teaching for a long time certain things, when some novelty comes in, you wouldn't want to change. So there are sometimes the agenda might go in different directions. So this is also an aspect of reality. And then Max Planck, one of the biggest paradigm shift scientists, he said that a new scientific truth does not triumph by convincing its opponents, but rather because a new generation grows up that is familiar with it. So novelty in, say, the scientists and the society individuals that we are, our society, the seniors keep things going and the juniors or even the seniors can bring novelty. But the point is this exchange must be healthy and this change must be in a metered and controlled manner. So with that, I move on to the next slide. The philosophy of science per se rests on negation. So the way in which Kepler came to the elliptical orbit, very classical example, wherein he used a traditional thought process, the Tycho Brahe's one, and his observations, meticulous observations of Tycho Brahe, Kepler said that it's not the circular motion, but it is a elliptical motion. So what does a scientist do? A scientist basically explains natural phenomena. That's the role of a scientist. A scientist explains natural phenomena, and an engineer builds the world. Okay, so slight difference, but nowadays everything is same, as Professor Soman said. Now imagine that you have something, you have a phenomenon. We try to explain phenomenon. So now you imagine that an ant goes, there is a ant moving around, and let's say somebody puts a hypothesis like the ant is moving in a circle. Now a circle has an equation 2 pi r defining its circumference. So if you want to ratify or question the theory, what would you do? Hypothesis? You would collect data points in real space and time about the ant's motion. So suppose you collect data points here, 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 and here, and then you fit it with the equation of the circle, it fits perfectly. But what if you collected here, here, and there, here, and the ant went like this for a transient period of time? Then you would say it goes in a circle, but then you would not be able to cover this. What if you collected data points here, 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 and continue to fit it in a circle when it is not at all anything like a circle, or it is completely different? It doesn't even have a continuum. So something like that. So in here, I wanted to stress on one thing. Science, unlike in math, you can prove. In science, the progression is through disproving. So the philosophy of neti, not this, not that, that is very contextual in the process of experimental science to explain things. All right. So with these kinds of disclaimers, I go to the next slide. That is here. Even in math, when you try to, just by giving a mathematical fit to an experimental observation, see, you got a few sets of points and you can fit it into with equations with very good statistical fits. It doesn't mean a thing. How it explains your call is through mostly by subjective calls, because you, even with mathematical fits and all that, it might not approach the reality sense. This philosophical aspect is very important for you to understand and grapple. You see the picture here. Have you seen this before? This is MCHS waterfall. Now you see, you can draw like this. You can explain at one point the water always seems to go downhill, but yet you know that there cannot be a perpetual motion like this. But we can draw it. So visual seeing might not be there. The mathematical fits might not be there. What is needed is you need to keep on asking, doubting, questioning, disproving, and all that to arrive at the truth in scientific explanations. In this symposium, fundamentally, we will deal with a wide variety of aspects of life. For your sake, I would like to tell you that I asked myself the question, what is the most important thing you can think of? I think it is life. Because of life, you can think of anything else. So life is the most important thing you can think of, and life affords us thoughts. Thoughts sustain life. So with this circle, the most important thing that you would need in your life is oxygen. So how or why do we use oxygen is the question. In the schools, we have seen that, I've taken a school notebook. We have been always taught that respiration is very different from combustion, as is written in this notebook. One, combustion is uncontrollable, and respiration is a controlled process, and things like that. And when you come to college level, you are taught that combustion and respiration are somewhat similar, but in the cells, the organic compounds, like what Sir was saying, or what Ravi was saying, they burn and they produce heat energy here. But in the cells, they produce a chemical energy currency called ATP. Like you go out and you procure your things using money, the cells apparently do it with this. All other aspects of this reaction is very similar. Now you see, now when you come to university level, when you do great higher level of research, you come to this kind of an idea where oxygen serves as terminal electron acceptor sitting at a protein called cytochrome oxidase bound at a heme to make water. It serves as, that's the role. What does the modern scientific world think? It thinks that oxygen is needed to make water at the mitochondria, and that serves as a terminal electron acceptor. Oxygen accepts 4 protons, 4 electrons to make water. Some of you might not really understand or appreciate it at this juncture, but over the period of these next 3 days, we will argue that oxygen is not needed to sit at one place. Oxygen is needed to make this crazy kind of species, diffusible reactive oxygen species, on its way from triplet oxygen all the way. The triplet oxygen is what you see in this air that we use to make water. This is why we need oxygen, and this is what contemporary scientists regard as dangerous things that lead to cancer, aging, death, and all that. What my argument is that we shall challenge these ideas that is currently taught, which if people had heeded, it could have saved many lives even in the COVID pandemic. All right. So I would like to play some video here at this juncture, which will show you what currently oxygen does. You see, it's very orchestrated, very deterministic, like what Professor Ravi was saying, very coordinated, very systematic. That is people. Let it. So this is from a textbook, the most famous textbook, I would say, for the next part. First is, yeah. So you can see it's all very deterministic, like what Professor Ravi was saying, and it requires multiple phases. It requires gradients, very sophisticated motors, all that. All right. So you saw that basically this idea, multiple Nobel recognized idea, I mean, more than half a dozen Nobels have been directly or indirectly on this, and which is currently taught in textbooks. It's what is going to be questioned. And the applications that come afterwards are going to explain a wide bevy of reactions and processes, and it enhances our understanding. So that was actually recently in December, I published along with other collaborators of mine, my group people. We published it in American Institute of Physics. It was founded by some very senior, very well-known names. Here you can see, Langmuir, Debier, Pauling, all that. So they founded this Journal of Chemical Physics. I had submitted this reviews, 25 years pursuit review in this journal. So it was reviewed and accepted, but then the editors decided that it would be published on their online portal, AIP Advances. So this review challenges multiple Nobel recognized ideas that are currently taught, and it thousands of papers in top journals like Cell, Nature, Science, PNAS. So with so whatever we know now, significant part of this world, have come from the greats that you see here. Can you mute the other speakers? This is the most famous, most famous. Yeah, I think it's Laurent. No problem. Let him be there. No problem. So this is perhaps the most important conference in 20th century. It's 1927, Fifth Solvay International Conference. You can see all the big names in the physico-chemical world. So what I say is, perhaps this symposium, even if not attended by big names and all that, in terms of contribution to science and our understanding of life processes, this is, I would say, as important as that. Thank you. So yeah, the kids who are sitting there back, you will remember this. After 20-25 years, this you would remember. All right. So this was held at Belgium. So with that Belgian connection, now I would like to hand over the presentation for the remaining part of the forenoon. I mean, it's already become afternoon, 1 o'clock. We are in good time only. This was planned. So next one is the when Laurent Jaeken, the esteemed colleague and collaborator of mine, will take off. Laurent, are you there?
Yes.
Okay.
Can you hear me?
Yes, we can hear you fine. You can use the slides at your end, or I can change it from here. Which one do you prefer?
Yeah, do it from there.
Yeah, I will do it from here.
I will say next slide and then you do it. Okay.
Yes. All right.
Yeah, we start. I'm Laurent Jaeken, and I am, during all my career, a hunter of errors, big errors in science for almost 50 years. And I was hunting for, I learned from this hunt that people were forgetting an important substance existing in cells, and it was water. And water can be in all the many different shapes and properties. And in the traditional view, which you find in textbooks and which is told to you in classes, water is said to be normal water. And I learned that this was a very big error, because if it is normal water, they will use the laws of ideal solutions and apply it to cells. And in fact, cells are a coacervate. And then I wrote a book in 21, 3 years ago, in which I collected all my hundreds of arguments. And probably Murali, Professor Manos was reading it, and he took contact with me, and we started to collaborate on this project, because there is another very important product in cells which is not studied, and it's oxygen. Both water and oxygen are very strange products with very strange properties. And this will be my talk, to see what Professor Manos actually discovered about oxygen, and how his findings are really entirely different from the classic view, which is falsified already for many years. And this falsification, he put another large set of arguments against. So in order to understand that, you should first know very well what is the classic view. So next slide, please.
Sorry. Thank you. Laurent, you are on. Connect. Did you close it? Laurent, are you there?
Yeah.
Yeah.
Cell theory, it means that all living beings, big and small, they are all composed of cells, and that's the structural and functional unit of life, which each cell can, in principle, do everything what a cell needs to do, what is typical of life. They are bordered by a membrane, and there is an inside, and that makes a common unit, which at the time was called protoplasm. It's the two together, they work together. Next slide, please. Next slide, please. Yes. The other important concept is called the central dogma, and it says that the information flow is from DNA, which can replicate it for the next generation, but which can be transcribed into RNA, and the RNA gives the information to the ribosomes to make the proteins. In this lecture, we will look to the proteins, not to the DNA, to the RNA. And what is typical for the proteins, it is that they are the real-time executors of all functions. The genes, they work on the long term. They cannot act at instant. And particularly with respect to oxygen, all reactions with oxygen are the fastest they are. So we focus on the real-time function of cells. Next slide, please. An important point is that enzymes can be considered as very, very small reaction vessels, which you use also in a laboratory, but much smaller. They are so small that they only take one molecule at a time. And therefore it is very, very accurate. There will be no side products formed because the reaction products, they fit into the active center. And that's specific, and there can be hundreds of products around the enzyme. The enzyme will pick up one of them. It's selective, it's specific, and by this you get control about everything what happens in a cell. And then going back to the central dogma, the DNA decides which proteins will be made. It's also deterministic, and everything seems to be controlled. And then there are some pumps in the membrane, and they control what will be inside in the cytoplasm for solutes and ions and all this. And it starts, they regulate everything, that it is the internal medium is very constant. And then all the proteins which are inside, they have an easy life. They shouldn't adapt from moment to moment to the outside conditions, because inside is safe. And one thinks the pumps, they arrange that in the membrane. And all this determinism, I tell this, people at this moment think that all enzymes work in this way, and that's not correct. There is among the enzymes a special group which was called with the murburn concept, it was called murzymes. And they work entirely different. It is just a new innovation. It was a revealing, really, that some enzymes work different, not according to the scheme, not according to this Michaelis-Menten kinetics. Please, next slide. So we should go beyond the classical view of life, and it was said already, in science at this moment, there is a huge tendency towards obtaining a consensus, and that's actually not a good thing. Science is not democratic. There can be some brilliant scientists who think differently, and they might be right. But if they don't be heard, yeah, then it's wasted, their genial ideas. And I put here some very, very important persons. There are other, of course. They didn't got a Nobel Prize, but they said important thing. And for instance, Beutner in 1914 already said that at a membrane or a fatty surface, like a membrane, for instance, it works like an electrode. That's the idea which now Professor Manoj is working out. It works like an electrode. Bauer, he said, work output by cells, the energy from it does come from the structural energy. It is order, it is low entropy, or it is low entropy, you can generate work. Ling here worked almost from 1945 till his death a few years ago. He worked on the concept that the cell is a coacervate and not a diluted solution. And also the other two, they followed this idea, and I personally also followed. And Hiroshi Tamagawa also followed the idea that a cell is a colloid, and also Manoj is of this idea, a cell is a colloid, and there is direct proof from it at this moment. So the classic view, they still continue thinking that inside the cell it is a diluted solution, and all their ideas are derived from it. We cannot continue on that path, impossible. Next slide, please. Next slide, please. Next slide, please. Hello. Yeah, thank you. Now, the central idea, classical idea, is that of pumping. And here are two examples of it. One is in the mitochondrion or the bacterial membrane, mitochondrial inner membrane. And it is a thing which was shown already in the video. It is NAD, the redox complexes are pumping protons to the outside. There is generated a proton motive force, which is the force of a gradient. And then this force is used to synthesize ATP. You have the same in the eukaryotic cell membrane, where the pump is thought to control the inner composition of the fluids inside the cell. And this needs three things. First, you should identify that the solution gets changed, and then it should be compared with what it should be, and then the difference should be compensated by pumping ions inside or outside. From this scheme, the two first points, identify and estimate, there are no data on it, no clear data on it. Why? Why? So perhaps because it all works very differently. Next slide, please. We will, no, one back. Yeah, chemiosmosis. A student can calculate in a few minutes that the whole idea of Mitchell, for which a Nobel Prize was given, is just invalid. He takes the volume of a chloroplast. He takes the concentration, which is near neutral, of the protons. He uses the number of Avogadro, and he finds that there are only 9 protons. And for a mitochondrion, which is somehow smaller, there are only 6. And for a cyanobacteria, which is still smaller, can be smaller than 1 micron in diameter, there is but 1 proton. Now tell me, how does 1 proton be divided over several thousands of redox complexes to be pumped out and produce there a huge proton motive force, which should be sufficient to produce ATP, which is an endothermal reaction? How is this possible? And how they will see the protons? They are that small. So just this calculation, I once was sending this calculation to Science magazine, and it was rejected because there were other more important articles to be published. Yet it's the definition of pH. It's about free protons and not bound protons. Next slide, please. Next slide, please.
Lauren, you have muted your phone, microphone. Lauren?
Yeah.
Yeah, now we can hear you. Okay.
Yeah. So the electron transport chain, you saw it in the video. The idea came from Kalin in 1939. He discovered the cytochrome, that there were several cytochromes. And then he was put in front of a very big question: how should it work? And there were 2 possibilities. Either they worked in series, that the electrons are translocated from one cytochrome to the next and finally arrive at the terminal electron acceptor, which is cytochrome oxidase, and only there is a reaction with oxygen. That was one possibility. There was another, that this redox complex works in parallel, each complex doing its own thing, and each of these complexes reacting with oxygen, liberating the energy, and then in some way that energy is used to make ATP. That were 2 possibilities. And he was in front of a very big dilemma, and he found a very, very, very nice argument to say it is a serial. like the one in the video. The reason was that if each of the complexes would react with oxygen, you would produce reactive oxygen species, and they are terribly dangerous. That was, and from that time on, this idea continued up to now. And that should be stopped now, because things are much simpler than this. One of the big values of the murburn concept, it is that it is a simple concept. And because it's a simple concept, it could be originated during early evolution. And if you think back to the video and you see how complex was this serial electron transport chain, and how complex was this mechanism of Boyer, of how the ATP synthase works, then you may really ask the question how evolution could ever make something like this, because it should be perfect before it can start to work. And actually, our calculations of protons, they even say that it doesn't work even now. So we should look for something simple. And if you see here, down on this slide, the oxygen takes one electron and you produce superoxide, and there is a lot of energy coming out, much sufficient to make ATPs, really. Next slide, please. Here is the idea of Boyer of the ATPase, the ATP synthase. And how it works you saw on the video, but here on the left side, you see an analog. It is a press for fruit, and the fruit goes in, you turn the rotor, and the juice comes out. Now what Boyer says is just the reverse. You put the juice in, turn in the other direction, and you get again the fruit. That's what it tells. Impossible. Next slide, please. There are other really slashing arguments to say that the whole idea of the serial electron transport chain and of the proton pumping and of the Boyer mechanism must be totally invalid, because one we told already, in cyanobacteria, they are aprotic, yet they can respire and they can photosynthesize, and in some of them, the membrane where it happens, where the respiration takes place and where the photosynthesis takes place, is the same membrane. So how should this proton current go? Inside, outside? Well, it's clear it cannot work. There is another slashing observation. It is erythrocytes in mammals. They do not have a nucleus, no genes, no mitochondria. They have glycolysis to produce the ATP they need, but it has been shown that is insufficient. So how they survive? It will be taught in one of the other sessions that hemoglobin can bind ADP and can produce ATP. Then there is a third thing, that plants, they do not have a sodium-potassium pump, yet they have a ratio of potassium over sodium, which is higher than 10. How they arrange that? Next slide, please. Really much of traditional science, I should say, the first chapters of textbooks, the first chapters of an initial course in biochemistry or cell biology should be changed entirely from new concepts, which are valuable, from the idea that the cell is a coacervate and that water is important, and from the idea that oxygen is important and do other things than just only react with cytochrome oxidase. Excuse me. What the murburn concept of Professor Manoj realized, it solved the mystery of ROS. The mystery, I mean, one knows that at high concentrations ROS is dangerous, but one does not know what the ROS does at low concentrations. And those functions are really astonishing. It's new. There is a new mechanism for redox proteins. And with a new kinetics, which is not a Michaelis-Menten kinetics, there are new ideas about those response relationships, which are very interesting for pharmaceutical industry, which are busy with medicaments, how they work. Some medicaments surely work differently than generally thought. You all know the big maps of Boehringer Pharma, which are on the walls in laboratories where you can see the overall reactions going on in cells. Large parts of these charts should be remade once you know what is murburn concept. There should be a new view on the homeostasis of the fluids, because it does not work. It is not kept constant by the pump. It works differently. There is truly an entirely new bioenergetic, how cells are energized. And by energizing of cells, it starts with redox reactions. And all these redox reactions, they work according to murburn concepts and not to pumping ideas. There are new insights in electrophysiology and in motility, because one of the end products of the murburn reactions is water. And water puts the cell under pressure if it is constantly produced. And with this water under pressure, you can drive hydraulic mechanisms. It's something entirely different than things working on pumps. And of course, the murburn concept tells you what could have been the very early origins of life, because it can give some ideas about, because the mechanism is so simple and does not use. How could ATP be made before there was a membrane? That's the point. And also glycolysis, if you look to the details of the enzymes with all the coenzymes which are involved, these are not simple reactions. The ROS reactions are simple, it's just basic chemistry. And we can believe the murburn concept because every detail is thermodynamically sound. All things are exothermal. There are no endothermal points, like, for instance, making ATP. The reaction to make ATP with the murburn concept, it's an exothermal one, because not only because it's a reaction with a reactive oxygen species, and there is not only water produced, but there is also hydrogen peroxide produced. It's a completely different. So the idea how to make ATP, it is now just an exothermal reaction. Everything is exothermal. It is kinetically sound because these reactions are very, very fast. And the very fast things, only the reactions with reactive oxygen can explain that it goes so fast. There are very nice structural details on the proteins, which really indicate that there are in the structure of the proteins points which structures which point to reactive oxygen reactions. and it's evolutionary sound. So why not? If about such important things, there are two views, and you are an instructor giving lessons, or you are a writer of a textbook, honesty declares that you should put or tell to your students that there are two explanations and not one, and that there is not only a consensus view. And you should give the arguments for the two arguments pro and arguments contra. That's the only way to go further with science. Next, please. Next slide, please. So we come now to the basics of the murburn concept. I tell them so that you can better follow the next sessions. Next slide, please. Of the fundamental forces which exist in nature, where you have gravitation, electromagnetic force, and then some forces inside the atomic nucleus, which are not that important. Life uses the electromagnetic force, and life is driven by electromagnetic radiation, visible light by photosynthesis, in the early days also by UV, and it's also driven by some infrared energy, which keeps us warm. Cells are made of molecules and ions. And so if we think to chemistry, how chemistry works, you have redox reactions and you have others, and the other, they work by the inductive effect of Lewis. But in both cases, what happens, it is that there are displacements of electrons. If you think about an electronic circuit, if you want to understand an electronic circuit, you have to look where the electrons are going. Here also, if you want to understand how a cell works, look how the electrons are going. In case of respiration and photosynthesis, they go to reactive oxygen species. Because it has been shown that everybody knows that each redox complex in respiration and photosynthesis, it produces reactive oxygen, and one is annoyed by that fact because it shouldn't, but in fact it is produced. And if the respiratory activity is higher, there is more of it. So there is even a direct relationship. Yet they continue by telling this classic stuff. They just write over one from another. That's the point. One should always look to the alternatives. And so, following the flow of the electrons, that is the primary activity. That is what life does. There, all power should come out. And it will be shown in one of the sessions that in neurons, one always thinks about the Hodgkin-Huxley, that there are ions moving across the membrane, but there are electrons which are doing the thing. That's the primary point, and there are ROS produced, and that can generate a potential. So it's all quite different from what is generally told. Next slide, please. This is the other important point of the murburn concept. At the left side, you see a classic enzyme catalysis in two ways, the enzyme, and down at C, you see the electron transfer enzymes, how they work. And they work by the mechanism of primarily there is an active center where the substrates attach and are selectively recognized. in the murburn concept, it's entirely different. You have the protein which donates the electron to the surrounding fluid where is the oxygen. And there, the oxygen, I should say, it's not only in the fluid, it's also in the membrane, because it's a rather apolar product, oxygen. And so it can be as well in the membrane. It is surrounding everywhere. And then the electron is donated, and there is an immediate reaction because that's a radical reaction. And the radical reactions that are the fastest there are. And it starts to react here, the ROS, and as a radical, it immediately starts to attack other products in the surroundings. And for instance, if there is ADP there, it will attack it and make an ADP radical. And if there is phosphate, it will attack it and make a phosphate radical. And then these two radicals can react with each other and you have ATP. Just simple like this. Easy for evolution to get there. And so you have there the enzyme catalyst by means of ROS, like for instance in chloroperoxidase, which can attack very many different compounds. Yeah, ROS can react with many things. And you have done the case of the redox complexes here below. So it means the name murburn comes from burning. It's a burning reaction. It's the same as in a flame or in a combustion engine. And the mur, there are different possibilities from where come the mur. It can be from molecule, unbound ion radical reactivity, or it can come from mild, unrestricted, free in the fluid, redox catalysis. Or it can come also from murt. Murt, it will, one may ask the question why we don't see this flame inside our bodies. Because the reaction chambers are very, very small. They are small cavities inside our redox complex, we are known, or spongy regions, or small liquid-filled regions between subunits of multi-subunit proteins. It's murt. There are millions of murt small reaction chambers where the ROS reactive, and of course some may escape from it. And if the concentration of ROS becomes too high, there can be danger. Next, so basically it is a simple chemical engine which is driving our life. Next slide, please. Here it is put in a shadow. You have the enzyme, the murzyme we should tell, because it's worth to give a special name for this special type of enzymes called murzymes. They give the electron, the ROS is produced from the oxygen, and there are ions in the vicinity, there are substrates, and all this is interrelated by chemical equilibria and eventually by adsorption and equilibria, which are also simple physical-chemical equilibria, and there are products. There is an output of products, but there is something else, another output, not yet shown on this sheet. If you have a membrane, and on one side, take the inner mitochondrial membrane, and on one side, you have donation of an electron to the fluid, and ROS is produced. Many of these ROS are anions, and there is also hydroxyl ion produced, and they are negative. And so you generate the negative potential, not by putting protons to the outside. It's just by, it's an electrode potential, like on an electrode, just the same. And this is a mixture, complex mixture. And what happens there is controlled by thermodynamics, by partitioning. Yeah, I should explain. The anionic radicals are very polar. And near a membrane, there is polarized water, which has low water activity, and they will be there. The oxygen will be in the membrane and in the fluid. So there are partitionings, and that is, according to me, the point which should still be started to be studied to get a deeper insight. Concentration, stabilization, proton, spin locks, all this is equilibria which control what happens there. Next slide, please. Now, the murburn concept really explains very many different important observations in biology. First, we can take as an example the chloroperoxidase or the P450 oxidoreductase in liver. The big problem with an active site mechanism is that these enzymes can oxidize so many different substrates. How would this active site arrange that? But if the reaction is done by immersion outside, yeah, the ROS will attack everything there. Here, you can have different type of reactions. Because the ROS is able to do all these kind of reactions. I just told about that the anionic ROS, they produce the potential. And also for the cell membrane of a eukaryote, there is direct evidence that the ROS are involved. And there is, in most experiments, I will tell you that the oxygen is one of the forgotten molecules. There are almost no experiments done where an experiment is done under air with a control under nitrogen or argon. But if you would do that control, you would immediately see if oxygen is involved. And it is involved in the transmembrane potential of the eukaryote membrane. So the mechanism is entirely different from what one generally thinks. The reactions with ROS in fluids, it is just simple random chemistry. They are stochastic, they are not deterministic. It's an important point. And the stoichiometry can be variable because one never knows if this or another product is, for instance, by hazard or because it was given as a medicament, is not introduced in this reaction fluid there. And it has effects, and therefore there are side reactions, and the stoichiometry is not really one-to-one. And then we come to the kinetics, and this is an interesting point. If this chemistry is done by reactive oxygen, it can be very rare effects. And if you make the kinetics, you will see it immediately. Curves, not a Michaelis method, but curves like you see them here, are typical kinetics. And you can have another dose response effects which are very strange, that the same product at low concentration is an inhibitor and at high concentration is an activator, or even the reverse, that it is an activator at low concentration inhibitor at high concentration. And I take two examples, much used in respiration investigations. Dinitrophenol. We used to apply high concentrations, and then it is said to be an uncoupler and an inhibitor. And the same is true for valinomycin, another uncoupler. They used to use high concentrations, but it is known since the 60s that when you use the same products at concentration 100 times lower, then the same products are activators. It just tells one thing, that these substances do not work by blocking this proton transport through the membrane, this not existing protomotivores. No, they just enter in this redox activity with ROS, and there the concentration matters. And many medicaments are in that case. Next slide, please. So the murburn reaction is shown here, the upper part of the picture, NAD and NADH and oxygen. The NADH by the enzyme gives the electron. You get the superoxide. The ADP is present. The anions are there. There is a negative potential, and the PE comes in, and you get ATP plus water plus peroxide. It is an exothermal reaction. And the protons are generated in the cytoplasm, because in the cytoplasm, ATP is used for work, for instance, muscle cell for contraction. It is hydrolyzed there, and this hydrolysis reaction in most scientific articles and even in textbook, it is written in an incomplete way. They say ATP plus water gives ADP plus phosphate. It should be ATP 4 plus water gives ADP 3 plus phosphate 2, excuse me, and not plus, minus, and phosphate 2 minus plus 1 proton. So there are protons produced continuously in the cytoplasm, while the mitochondria is nearly aprotic. So they come in, but they have a problem, because the membrane is the surface of the membrane. There is polarized water adsorbed to it, and the protons cannot pass through it. So what the ATPase has to do, it is if you hydrolyze the ATP, then the polarized water will be depolarized, will become something between normal water and maybe even hyperfluid water. And then the protons can come in, and the protons control this reaction mixture. So the ATPase is the controller of this chemistry. Important function, but a different than generally thought. And then there is the other thing, that the cell is not, the fluid in the cell is not a diluted solution, but is a colloid. And all these things together in a colloid, and also with a redox reactivity, this can fully explain the strange concentration differences between the inside and the outside. There is one big similarity between the murburn and the colloid nature of cells, these are two theories which do not need pumps. So we can really abandon all this idea about pumps. Next, please. Next, please. Next, please. Please, next. Here you see how important is the murburn concept when one looks what it all does. It explains respiration. It explains photosynthesis, and the whole mechanism is different in both cases. It explains thermogenesis. It explains the Cori cycle, because in the Cori cycle, you have reaction from pyruvate to lactate, and from lactate to pyruvate. It cannot be done by the same reaction mechanism, impossible. You have the drug and xenotic products, metabolism, and dose responses, which are pharmaceutically important. There will be a new view on how the visual cycle works, because also there ROS is implicated. There will be some sessions on electrophysiology to tell you that the things at the membrane and the potential is also completely different. And a very important point and a strong argument for murburn is that it is the only theory which can explain why cyanide works at such a low dose and so fast, also because the ROS are inbound. And there will be a section on the very, very early origin, prebiotic origin of life. And I can tell you already, without ROS, there would even not be amino acids. So you see, it's a really very broad science, important, very important science. It is perhaps the biggest paradigm shift there has been in biology. Yes, next slide, please. Here you see, I started with telling about the cell theory, the central dogma. There is a third important pillar to understand life. It's the murburn concept, is the flame of life. Because basically life is a redox activity. You can see it at the molecular level, what happens, which reactions go on. You can see it at the macroscopic, let's say, cell level. And then you see, yes, carbon dioxide goes out and water is produced and has to go out. And there is heat produced because the reactions are all exothermal. And the fact that there is heat produced, it makes that it is the drive of the chemical reactions. You should have something which produces heat, and this gives the direction of the whole system. And you see it is actually the same like in a chemical engine. Next, please. Next, yeah. Here I will not detail this, but you have an overview. You can look at your own. Here you have the classical view and the murburn view, and what are the difference on all these points. And these are the different criterions. It's a really, really long list. Next, please. Here, it might be a delicate question because important people, important concepts given in textbook, given in initial courses on cell biology, cell physiology, biochemistry, things have to change. Hill said the cytoplasm is a dilute solution. It is not, it's a coacervate. Kalin said it was a serial electron transport. No, it's a parallel. Dean said that membrane proteins pump ions. Hodgkin and Huxley say that the cell potential is due to a system with pumps and voltage gated carriers. It's much more complex than that, and oxygen is involved. These people, they described the classical retinal visual cycle. It's another one. The flagells of bacteria, it's a rotary motor. No, it cannot work as a rotary motor. Just like also the Boyer mechanism, the rotary idea, it is not sound. Peter Mitchell ischemios doesn't work. Also Boyer's complex thing doesn't work. Then there are SCU, electrogenic sodium potassium pumping. No, the potential is not generated in that way. Then you have two others, Neyer and Saxman and McKinnock. They designed methods here to study single ion channels, but they also used a blank which consisted of a silicon membrane, and there is also a potential. And here it is the same. There should be a blank. And why not try this and this under air and under argon to see if there is the reasons there is a ROS implicated or not. So we cannot trust maybe trust the methods, but not the explanation given to it. So you see, really, this is a huge revolution, and much should be changed in courses and textbooks. Next, please. I will stop with this picture. It's about reactive oxygen species. We have now learned, and we can first look here, a knife is a dangerous thing, but you can use it to prepare meals which are needed to continue your life. And here it is the same. The ROS is a dangerous thing at high concentration, but it's something absolute needed, and it is the most one of the most central reactions in entire metabolism. It is the coupling factor and production of the electric activities. So I will give the word again on someone else. I don't know who will continue, but to again give the philosophical application of all this. Thank you for your attention.
Thank you, Laurent Jaeken. Thank you, Laurent, Professor. I hope you will stay on. I'll just finish the last couple of slides here. Here you see, it's just an aesthetic perspective that ROS are dangerous, reactive species. Now, when you go to a kitchen, you will find cutting boards, knife racks, gloves. It doesn't mean that knives are not used in kitchen. When there are antioxidant enzymes in the cell, it doesn't mean that ROS are bad. It means that they are there for a way to deal with it. So just as using a knife is dangerous, but without a knife, practically you can't get anything cooked in in a kitchen. So basically, the murburn concept is sound on energetics, kinetics, mechanism, structure, distribution, evolution, probability, Occam's razor, all that. But only one problem is aesthetics, where it comes to acceptability. The tradition of seeing something as bad has been something has been a barricade. All right. So the philosophy is something like this. You are something that's burning. Believe me, you are. When a cloth is dipped in oil and it is set on fire, what happens? The oil burns first, but then cloth also burns slowly. That's what is happening to you. The thing that powers you, that sustains you, is that burning process. What Professor Raveendran said, it's all deterministic, everybody was saying it like that. That is important, that enables us some choice. But fundamentally, we are ultimately not blessed with that many choices. The fundamental powering logic is chaotic. We have emerged from chaos. To chaos, we must go back, because that's the burning that happens. The wick will get charred. It will get powered, but it will have to die. That's the reason we die. But then that's the philosophical part of it. That is the ROS, or the reactive oxygen species, just like Dr. Jekyll and Mr. Hyde. It has two properties. This is the Batman's comic, Scarface. I used to read these kind of things when I grew up, so that's why I put. So order from chaos and chaos from order. So life giver, life taker, both. It's the fundamental thing for which we need oxygen, is that it is used to make the ROS, reactive oxygen species. That's the reason that we need. So to put it short, the classical biochemistry sees diffusible reactive species as toxic. So, but then murburn concept sees that enables effective charge separation, the most fundamental thing about moving electrons or making new bonds or new moieties, right? So, and that enables the cells to work as simple chemical engines. So these are, marketing words put there. ECS, effective charge separation. SCE, simple chemical engine. So this idea will get sold. It will get easily, it will get absorbed in the society. So basically, the murburn. Now you see what happens is, if I go back to the earlier slide, one of these slides, see here, the thing is murburn concept. What people have been seeing is mostly this. So you imagine, suppose now a drama is being played on a stage. Now the drama can also be qualified by some, chance lightning, some the shaking of the stage and things like that. So that part, the whole stage is murburn concept. That has evolved. Central dogma and cell theory has all come because of that. So you can see at molecular level, at macroscopic level, at the conceptual level, the roles of murburn concept in the way in which it functions from the molecular to the macroscopic to this whole organism level, tissue, organism level, all that. So I wanted to say that murburn concept is a fundamentally ubiquitous thing that happens all everywhere. Molecules and ions, and they interact. Chance radicals are produced. That's what it is, murburn. That's so you would see that in our ancient texts, we have called the process of metabolism, digestion, assimilation, and all that. We call it dahanam. Dahanam, digestion. Dahanam is burning. So we have called Jataragni, Kayagni, Dehagni. This was brought to my attention by Sai, our our Chief Innovations Officer. I realized that it's not that very innovative. Ultimately, our ancestors were already seeing it like that. But then we have put some, my group and I and my collaborators have put some, details that is there. See, Charaka Samhita says, Shante agno mriyate. When the fire dies, you die. So that Laurent Jaeken was telling me that even in the West, they used to say that. So it is not necessarily that all that is ancient or all that our forefathers said, all that is wrong. No. That's a very bad way to look at it. We should see, we should understand, we should try to see in what context, in what perspective. So the homeopathy aspect also that Professor Raveendran said, sir also, Govi sir also said, Govindankutty sir also said, many things we have to see with a new perspective. So with this, I would like to put the first session to an end. Thank All those that have come, we have exceeded by about 15 minutes. It's okay. Now we can go for lunch. Yeah, we will have questions also. Before I take the questions, I wanted to first thank once again. Gratitude and respect is the founding principle of society. We human beings, we are not the strongest, the fastest, the most cruel ones out there. A tiger is, you cannot stand one-to-one with a tiger, no. But then tigers are hunting exactly like the way they used to do hundreds of years back. We have made strides. We are doing artificial intelligence, all that now. How? Because our seniors, they learned through generations. They passed it on. They wrote it down. There was a tradition, and that's how we build on each other. And then that doesn't mean that we keep parroting whatever they say. Some younger ones, some different ones amongst us, we get inspired. We think, oh, this might not be right. We make something new. But then the point is, this process can only be going on if we believe in this teaching process and the learning process and respecting and then maintaining that social decorum. And that's very important. And for that, the basic thing is that institutions must exist. Our seniors should be respected and they will support us. And you will see in the future, the younger generation, that you're going to learn the details of whatever have been saying or whatever Lowell has been saying or whatever Professor George Chaldakov or Sir all were saying. The crux, you have to have some basic approach to approach whatever we are saying. We will give you that. And I hope you young people, the majority, 75% of our participants are you people, younger students and scientists. So with that, we would like to take questions, but we can go on with the questions. But our respected guests, they might be getting late. It's okay? Can we take the questions? We can. Yes, please. Yeah, please. The mic is here for you so that it gets.
Thank you, sir. It's just further details will be surely presented by you in next presentations today, tomorrow.
Yes.
Just for the clarification and to understand that better.
Yes.
The murburn concept, the production of the energy or oxidation process takes place fully inside the cell, inside the membrane. And the classical has got some transformation or pumps through the membrane. Is there basic difference?
Basically, see, what happens in the classical theory is, now for the energy part alone, this cell must control inside. It must control outside.
Yeah.
It must have a membrane in between.
Okay.
That is the energizing process. Here, it doesn't. What happens is, with the advent of oxygen inside, the oxygen, because it pulls electrons, it is a random stochastic event. It separates, it strips, and there sets in a chaotic sequence of events. And that is, the whole life has evolved. The proteins, everything has evolved to capitalize on this fundamental energizing aspect.
Thank you.
Yeah, that's the fundamental thing. You got the you hit it by the nail. Yeah. That's a very fundamental argument wherein we can argue that the life process can control maybe inside. How can it control outside? And then another membrane. Multiple things it has to get for the life to begin.
Of course, you will be presenting further. We'll be understanding better. But later on, some questions will be asked because I want to listen to first and then ask questions.
Yes,
I think.
But this was primarily an overview of things. You know, Professor Laurent Jaeken, he put together a whole picture. Yeah. And so now we'll have multiple sessions. First, I will show you the chemistry that is coming up next session, and then we'll have some examples that my assistant professors, my boys will say, they're my boys.
Yeah.
So they're assistant professors now, but then it's like that. So
of course, many other theories can be explained on the basics of murburn.
Yes,
which you will be presenting.
Yes.
Thank you. Thank you, sir.
Yeah, thank you. Any other yes, you give the mic to Prasad, please. Prasad, Prasad. Yeah.
Yeah. So it was wonderful presentation by Dr. Professor Laurent, and it gave a very good overview of the whole things.
Yes.
So I wanted, for the sake of record, what are the three possibilities you mentioned about the expansion of MUR in murburn concept? That is one thing I wanted to ask. And what is the possible role of metabolomics and murburn concept? And how do you want structural biologists to look at murburn concept and design their experiments? These are the three aspects I would like to you to comment on.
Can you take the question, Laurent? That's the question is directed to you.
No. I hear it, but I don't see immediately an intake how to answer to it. I did not completely understand the question, I think.
Okay, I'll take the question, Laurent. I mean,
because there were three questions.
Yeah. I got it. Yeah. So basically, mured burning. First is mured burning. Mured is archaic English closed burning. Yeah, mured burning. Molecule. Yeah, cells are made of molecule unbound ion radical. So it's an interactive equilibrium. Unbound ion radical. Molecule unbound ion radical interactive equilibrium. And third one is mild unrestricted redox reaction. Right? Okay. Third one. Actually, there is another fourth one also. Yeah, no, there is a fourth one also. The fourth one is my name is Murali. When I was in 11th, 12th, I used to read all these reactions: Cannizaro's reaction, Kolbe-Schmidt reaction, Einstein's equation, this, that. I said, man, we are going to die, right? So we have our kids, we pass on our genes, physical continuum, work, we build, we teach. That's when your space mass attributes go, you should linger around as an idea. Murburn, there it is. Yeah, I burned. Yes. Murli burnt. Yes, you're right.
Yes, the problem, if I may,
all of us burn. Yes. Are you saying something, Laurent?
Yes. The point is, in fact, we really have a kind of flame inside, but one of the mechanisms to get it under control, it is to divide this flame over millions and millions of small flames, almost invisible, in very, very, very small volumes of, yeah.
So basically,
distance between subunits of a protein, that small are the chambers.
Yes. Basically,
but
when
the reaction mechanism is the same as in a flame.
Yeah, basically, when you burn in open, what happens is the molecules, ions, radicals, they interact, but the release is boom, like that, right? Here, the release is rather mild, unrestricted, but tempered. That's what the proteins do. That's the reason why we have a lipid membrane. Yeah, you're saying something. Give the mic to him, please.
So there is one very interesting phenomena in mechanical engineering.
Yes.
That is adiabatic process.
Yes.
So that adiabatic process is what exactly Dr. Laurent is right now describing about very small flames. They contribute huge, actually.
Yes.
So that adiabatic process, if we properly understand the philosophy, our life, whole metabolism is going by that process.
Wonderful. See, the point is now we are at the now the average lifespan was around 50 some few years back. Now it is going to be 60, 70. Now with people accepting Murburn concept and trying to understand life with that, understanding our eating, lifestyles, all that, our lifespan will easily get into 3 digits. If we have good food, of course. because the contamination is also going at the other end for production. So I predict that lifespan will significantly enhance with the good awareness of murburn concept. Yeah, basically, you see, I have stopped. Yeah, I eat only once a day, so I try to minimize. Yeah, so some people are saying, "Tang, cut from there." My coordinator. So I think people are getting grubby, right? You want to have food? Yes, yes. Your stomach is burning, huh? Yes. Anyone? Yeah, sir. Sorry. Yes, sir. Yes, sir.
Is it working?
Yeah.
Can you hear me?
Yeah.
Yeah. Currently, with my very limited knowledge, I have few questions to ask you and a comment also.
Yes.
The question here is that reactive oxygen species.
Excuse me.
Can you?
Excuse me 1 second. Here you are on a symposium. You are not supposed to do this amongst you. I will not tolerate it. This is not why this is. I will get mad. He is asking a question. You have to be listening. Please be nice. Yes.
Reactive oxygen species, we have heard a lot. It is valuable and is commercial also. Okay? Commercial in the sense there are a lot of industries, medicinal things based upon this. There are a lot of evidences also on reactive oxygen species, and very many, and even today, I have a feeling that reactive oxygen species is dangerous. Do you negate it?
No, not at all.
Okay, fine. That is enough for that. I understood later. We can.
It's like knife.
I can give an answer.
Like knife.
Yeah.
Everything has context.
So, yes. Okay. Let us.
Yeah. No problem. No problem. Yeah. That is a slide. Yes. Yes.
So the next thing is that it is not that okay to me, to my personal belief, that to associate such slokas and other things, what you wrote, okay, or like you, there can be very many people who can talk about Pushpaka Vimana, so many other things, and finally tell that our ancestors knew all these things. Okay, this is my personal belief that our ancestors knew all these things. So ultimately, this is what it is. That is, we are just reproducing the old knowledge. Please don't bring this concept to such a great meetings. Okay, I mean great in the sense.
1 second, sir. I heard you. Okay. 1 second. Wonderful. Yeah. You did not hear me out properly. I said very clearly that the burning idea, the idea of calling it as a burning process is what our ancestors said. And I said that I have explained certain things that can lead to the idea that it is burning. I never said that they knew the concept. What is my importance there? I never said. One, okay, two. Second, I like this. This kind of exchange should be there. Rational temperament should always prevail. I'm also for rational temperament. I'm with you, sir, on this. Second aspect is, it does not negate our ancestors' right to speculate or to think without. I did all this not necessarily with instruments and chemicals and all that. Many of the things that I could predict and all that was because of some amount of basic amount of common sense. See, when certain things are seen by people, they are taken for granted. Certain things, I retained a childlike curiosity, and my students and my collaborators also had that, and then we went together and we explored certain things. So when I said that, it was just to show that there were perspectives like this. These perspectives, even Laurent Jaeken said that 150 years back, it was even prevalent in the West, and they were completely kind of shut off. What I'm saying is, let us be a little open-minded, at least to some ideas that even our ancestors thought like that. That's all. What happens in scientific progression is, slowly some people get together and they completely block out a parallel system of thought. What we need is this kind of, whatever you did now, no, your opinion very strongly, your project. I like that. That is what we should do as scientists. That is what there is no point in agreeing every time. We should honestly and spread the rational temperament. I was not saying that they're all right and all that. Never I said that. Yeah.
Logics and some science maybe. But now we have a support of chemistry, physics, and so many others new sciences are being developed. With the help of them, we can now put it better than them. But the basic ideas can be taken from them also.
Yeah, you see, modern science, as far as I'm concerned, modern science is as good as ancient religion for me. When I talk to a doctor or many other liver specialists, when they try to prescribe drugs and all that without understanding what they do. So today's awareness of what is right is perhaps tomorrow going to be outdated. But if we remain open-minded and interact and try to see whatever supports us, we can quote it. Wherever oppose, we had put Gilbert Links and all those names. They were opposing something, and they were right in opposing. They were not really correct in saying some ways, but some of the things that they were saying. So some of the things may be right, some of the things may not be right. So we have to be very careful in this. Yes, and this kind of honest interactions and exchanges are what drives science forward. It's not mere parroting and following. It is the honest exchange of ideas that goes on. Yeah.
So is it possible to manipulate a murburn process?
Well, that is what I said. That is wonderful. Now, I'm a scientist. I explain. For me, the kick lies in understanding natural phenomena and explaining. Engineers and technologists will come now. They will, now, sir and other. We are already trying to, use AI methods to try to understand whether things are murburn, murzymes or not.
If we can manipulate the murburn process, so when we die, we could not control the murburn process.
Yeah, basically,
it will stop.
See, death can occur due to multiple reasons, right? The natural process of death is what occurs, through murburn. So if somebody puts a big hole in your heart, it's a mechanical damage. So, but then there are many cases wherein people go into coma and they remain in that vegetable kind of a state. And then something happens and they come to consciousness. I have a friend, Phil Penketh. I don't know if he has joined. He went into coma for almost a week, and he went around visiting some people in his place in UK. So these things are not really supported by science and all that, but you don't know. He is alive now. He says something like that. So what I am saying is keep minds open. You never know. Controlling murburn, the death process. There might be ways in which you can, like heart goes dead, the sinusoarterial node, that gives the electrical signal, right? That thing you can kind of give electrical signal and get it going. When the brain goes dead, we don't know. Brain goes vegetable, we don't know. Certain things we can, some people come back. We don't know. Certain things, now we are in the process. The neurological process is now going to be understood with murburn better, I think. We are at the stage where we are just going into it, Yes, sir.
So as a listener for 2 hours, let me explain. I won't take much time. Let me explain what I learned about murburn concept. Please. Okay. So the reaction happens with enzyme and substrate is there. Then enzyme substrate complex forms, and it reduces the activation. It reduces the activation energy, and the reaction happens. This is my classical concept about a reaction. Now, a catalysis, and this is what most of the reaction. Why I am feeling hungry is because of this type of reaction. When I have food, how it is chopping is because of this reaction. Now, what is the concept? Murburn comes. New idea. What you are putting here is what I learned from this seminar. That's what I wanted to tell. Maybe others can. Then you can add up to that. Okay. So what is murburn concept? This concept says that in this enzyme substrate complex is please add
reactive or diffuse oxygen species.
That's it. That's it.
Without that, your explanation is not correct.
Correct. Absolutely, sir. You got it. You nailed it. Basically, till now, you understand reactions in biology like this. There is a very see, I see Soman sir, I see Venugopal sir, I see Ravi. Now I know they are different. I see Gobi sir. I know they are different. How do I know? By their topology, their surface, their nose length, all that. Similarly, the enzyme reaction is supposed to work. That's what Laurent had put this thing. Okay, we are not going. He's there. So all the selectivity and specificity or biological process, it was supposed to be governed by what sir said. The active site binding, the topological, yeah, and thereby decreasing the energy barrier. Yeah. So basically this topological recognition, lock and key, which you have studied. Yes, Fisher's lock and key. That specificity at a topological level at a protein site. So that is not necessarily the only way the major reactions occur. The major reactions need not really occur exactly that chopping deterministic way. Some reactions can occur like this. You pour some water over a mountain and the water can come down in multiple ways. Multiple ways. It can come down in multiple ways. So this is what murburn is. It is not necessarily a deterministic electronic circuitry, but it is a stochastic multiple way in it. So you take a paper and you burn, right? Now that burning process, you try to recapitulate it with the second paper. It will not be identical. So how do two twins who have exactly the same genetic sequence, how do they think different? How do they behave different?
Sir, we can conclude the session. Next, other questions can be asked in the other
yes,
sessions.
Thank you, sir.
Thank you. Thank you.
Yes. Yes.
Thank you. Thank you.
Actually, murburn is nothing new. Molecule, ion, radical. Thank you so much.
Thank
Let's
you so
go
much.
for lunch.
Thank you so much.
Yeah, lunch is here. Down we go, and then just across the road. Yeah. We'll go. Yeah, hey, Laurent, sorry. Yeah, say Laurent, bye-bye. We are going for lunch.
Goodbye.
Thank you. Please put your hands together for Laurent Jaeken, please.
Thank you.
Wonderful talk, Laurent. I really loved it. Thank you so much. Yes. Thank you. Bye. അല്ല നിങ്ങള് people, students, you can see some AR, VR stuff we have. You know, you see our program in biomedical engineering and AI. We are going to use some state-of-the-art visualization techniques and all that. That is there. You can just have a look, okay? Please. But now you can go for lunch also. Lawrence, see you. Yeah, you can come later. First food. We have only 40 minutes for lunch. 2:30 the next session starts. Sorry. Yeah. Go ahead. Don't go elsewhere. Don't go sightseeing now. All right. Yeah. Thank you.